Okay, let's talk about vision by first focusing on the retina. All right, so the retina has these rods and cones, okay, in it, and it's composed specifically of a pigmented layer and the rods and cones. So when light enters here, this is the pupil, so when light enters the eyeball and goes all the way back to here, the pigmented layer of the retina is going to keep the light from scattering and just focus it towards the photoreceptors, the retinal receptors. And um, they're going to, so here comes the light. It is not scattered. It goes to the receptors here, and we have the rods and then the cones. All right, so the retina is a thin, fragile tissue carrying the visual receptors. Those are photoreceptors, rods, and cones. They face the rear of the retina, and they're shielded from the incoming light by integrating cells, right? These neurons are going to transfer from the receptors to the innermost layer of the retina, and then the axons, and then the optic nerve. So let me show you how this pathway goes in this image here. Okay, so light enters. It's called the optic disc. This is where light can enter. There's a space here so that the optic nerve can exit the eyeball. So your sclera actually covers almost the entire eyeball except for where this optic disc is located. So, all right, so light is coming in, and the photoreceptors of the retina are receiving the information, the wavelengths of light, right? And then they are going to process it and send it down these other cells, which then their axons converge into this optic nerve. Okay, so in a nutshell, right? Let's look at the photoreceptors here in a bit more close-up view of the image, and specifically here I want to look at the rods. So these so rods and cones both absorb light and trigger the nerve impulse, right? And rods on this side, there are many rods converging into one single nerve fiber and then synapsing out to the optic nerve. Here we have cones receiving information, synapsing just with one instead of branching out in this area, and then having each bit of information go into an axon of a nerve fiber. So that's going to create precision, right? This is going to create a vagueness because we have lots of information coming into one signaling firing out, and then here we have a piece of information traveling and signaling out on its own pathway. Okay, this is just a nice blow up of this actual portion of the photoreceptor. Okay, seeing the nucleus and then the synaptic ending here. So rods are deeper pigments. They're stacked. They're more light sensitive, so they're sensitive to light coming in. They're responsible for dim and night vision. There's no color vision here. The nerve fibers, like I said, converge to one, so multiple converging to one. The impulses to the brain, because of that, are less specific, and they produce an image with a general outline. They're predominant in your peripheral vision. Cones, on the other hand, are shorter stacked. So here we can see the comparison: longer stacked in the rod, shorter stacked here, require higher light intensity. So you need a brighter, higher light to come in. So when I was saying requires less light for the rods, it's that dimmer light can be received and interpreted. Here you really have to have a bright light in order to interpret it. Less convergent, one-to-one. The brain will then pinpoint the stimulus more accurately. So with cones, we get a crisper vision. They produce color vision, but only in bright light. Night vision is black and white. Color vision is due to different pigments in different cones: red, blue, or green. So in order to understand that, let's look at wavelengths, right? So depending on what wavelength the color or the light is coming in, right? So our brain is constantly taking vibrations or wavelengths and then interpreting them, right? So here we can see that there's blue green, and red cones that are going to be responsible for the various wavelengths. So if the wavelength is more around 400 nanometers, right, blue cones are going to pick it up, interpret it, and see it, versus if it's closer to 450 here, we get that green. And then the red cones are going to follow this red wavelength between the 500 and 700 way. So what's color blindness? Red and green are common color blindnesses. They're defective red cone pigment. It has the same light absorbing properties as the green, so sometimes those can get confused. Here we would have an insensitivity to red light, and then that's what leads to an inability to differentiate between red and green. Here is your colorblind test. So you can pause here and just take a look at it. And if you return to the video, I'll just read them out in case you do have some deficiency with the red receptors. And we have in this first box 58. We have in the second box a 1 and an 8. This is the letter E. And then I believe this is a 12. So maybe I have a little deficiency with these reds and greens in this tone, but I'm seeing like a 12 here or a 7. Wow. A couple years ago, it wasn't like this. I could definitely, I had no problem interpreting that. Okay. So look, this is like a nice view of a normal retina and what it looks like. Okay, so again, the retina is containing those pigments and the rods and the pigmented area and the rod and the cones. And then we have a retinopathy. Okay, so this is when due to excessive sugars in the body system, they eat away at the retina and they disturb the vision. This is called diabetic retinopathy. So it's a secondary symptom or clinical presentation or effect due to diabetes.